I'm Stephanie Countryman. I work with BioServe Space Technologies, and I'm going to talk to you pretty much what everybody else talked to you about, so space life sciences hardware and our expertise as a center. Next slide. Our mission at BioServe is basically to work with partners to conduct space life sciences research in the fundamental arena, the applied research arena, and also commercial research area. We do have a website. However, it's on the University of Colorado's website. We are a center within the University of Colorado. We're not a private company. We're not a for-profit organization. We're a center within the university. However, we've been conducting space life sciences research since 1987 when we were first organized as a center. It's the only thing we do. We don't do anything else except conduct. We build space life sciences hardware and then conduct or fly our partner research in that hardware on the space shuttle and space station. I've put a picture of Boulder, Colorado up here. So in case I don't say anything that encourages you to come and work with us, where we work should encourage you to come and visit us and potentially work with us. Except if you're an investigator in Hawaii, I just want to say that I'll go there and visit you. So, next slide. So what is our expertise? Well, first of all, developing research partnerships. We're very experienced with that. Almost all of the missions we've been on have been conducted in partnership with some other type of organization. We understand the agreements that need to be conducted. We do confidentiality agreements, material transfer agreements, intellectual property agreements, so we understand research partnerships. Also in our center, we have in-house research expertise. It's something that makes us somewhat unique as a space life sciences research center. We have the engineers, but we also have the scientists. We have particular areas of expertise in the musculoskeletal disuse arena and bacterial responses to low gravity. However, we have experience in a wide variety of research, including cell culture, tissue engineering, other microbes, small organisms, plants, and animals. We're also experienced in flight hardware and software development. I'm going to show you a little bit of our hardware in a few minutes. And then also conducting the spaceflight research. We are experienced in the planning and execution, and then all of the paperwork that needs to be done in order to fly your experiment to the space station. We're basically a one-stop shop. You can come to us, we can advise you on your science, and then we can do everything else that needs to be done to get you flown. And you would never have to go to another partner. We also do education and public outreach because we're located within a university. We utilize undergraduate and graduate students. We're a small organization, so the fact that we have access to those students helps us conduct and do more with less. Next slide. As I said, we're a small team, but we have extensive experience. The average length of time is greater than 10 years of our employees that are working with BioServe. And I also want to make a comment about these photos. Don't let the fact that we cannot take good portraits deter you from working with us. And also our lack of fashion sense. It's not so good. But we're really good at spaceflight research. So, next slide. We have a long history of conducting research in space. We've been on our payloads, and our partner research has been on over 40 missions on the shuttle, the space station, Mir space station. We've also been on Soyuz in progress, so we're very well experienced in this arena. We've done dozens and dozens of individual investigations, and I'd like to point out that we've flown on 40 missions, but 70 different types of payloads. And what that says is that we take your science and we don't just give you a piece of hardware and say, here's the hardware that's available to you, fit your science in it. What we like to do is we have a large suite of hardware, and maybe we need to make some small modifications to it so that we can help your science get the results that you want in space. So we don't try to just cram your science into the hardware that we have. We try to make it optimal so that there are restrictions with the hardware, but we want it to be able to successfully fly your science. And we've done molecular, cellular, small organism, plant, and animal studies. Next slide. We obviously, like everyone else, can do all the services that are required in order to conduct the research in space. I also wanted to make a quick comment. Somebody commented earlier about export control. all payloads that go to the space station have to pass export control. You have to analyze it and make sure that it can. But we can do that. We've done that. It's usually not a problem with life science research. Next slide. I only put this up here to make the point, which I think I've already made, is that we have scientists and engineers that can, our scientists can advise you on the biophysics and the biological aspects of flying your research in space, and then our engineers can talk with the scientists. Our engineers are very comfortable talking to biologists, and our scientists are very comfortable talking to our engineers. And that's a really important interface to be very good at in this business. So we are able to advise on that. Next slide. I'd like to show you some of our hardware quickly. We have a unit, it's an incubator, and it's on, we have 2 of them on board the International Space Station right now. And it's called CGBA, and I can tell you what the acronym means, but it really doesn't mean anything. So CGBA is how it's known, and there's only a few small group of people left that actually know what that acronym means. So it's an incubator, as I said. It has a freezer and a refrigerator configuration. It can go from -16 C to +40 C. Right now, the 2 units on the space station are in the freezer configuration, which is -16 to +40. We have 2 that will fly on the shuttle, and they provide +4 to +40. So we can provide thermal control for your experiments. The thing about CGBA is inside of it, we have all these small pieces of habitat hardware that go inside of CGBA. So we don't just have a big unit that then you have to figure out what to put into it, but we have all the little pieces of hardware that go into it, and then we can provide you something that's appropriate, say, for cell culture, but then also provide you with the thermal control. Next slide. I think I just covered that. We have 2, this is the technical data on CGBA, 2 on ISS, and 2 that regularly go up on the shuttle. Next slide. Some other hardware that I'd like to show you. This obviously is not all the hardware that we have, but this is also one of our center's workhorses pieces of hardware. It's called the Fluids Processing Apparatus, but actually we call it the FPA. It's basically like a test tube. It can mix 2, 3, or 4 fluids. It can fly in a wide variety of configurations. We have flown actually thousands of these on the shuttle and space station. They can fly by themselves, or they can fly in what's called a group activation pack, where 8 of these test tubes can fly within one container. 16 of these smaller containers fits inside our CGBA. So 16 times 8 is how many samples you can get. Because CGBA is thermally controlled, we also have automated, and it's smart. It can provide automation to our experiments as well, so we can either automatically activate your experiments, or we can do it manually via the crew. If it's done automatically, we can activate some. If it's done actually automatically or manually, we can activate some of the gaps at one point, and say 3 other gaps at another point. We can also activate thermally. So you can hold your samples at, say, 4 degrees C until it gets to station and you want to activate your experiments, and then we can increase the temperature of CGBA and then activate your experiments for a certain length of time. And then when they're done, you can terminate them or cool them down to maintain your samples until you can either analyze them on board the ISS or bring them back. Next slide. And this is just a picture of a gap that is automated. Next slide. This is just one configuration for a microbial growth study of the FPA. Next slide. We also can do yeast studies in it, where you can dry the yeast down on a filter disc inside the FPA. Next slide. We can do infection studies where there's actually 4 chambers, so you can have your cells of interest in a growth media, and then you can have an inoculum or a different growth medium in a second and third chamber, and then your 4th chamber can be a fixative. Next slide. We've done protein crystal growth studies in these. Next slide. Suspension mammalian cell cultures, where they're either suspended in the media or on beads. Next slide. And then we've done attached mammalian cell cultures, where they're attached to a cover slip. Next slide. And we've done insect development where you've put the pupa, or the pupae of the insect into one chamber, and then you can have media in a second, and then you can either have a plug in the third chamber or some type of fixative. Next slide. And these are all the different types of organisms, cells, and microbes that we've flown in these FPAs. Next slide. We also have an alternate configuration of our gaps, the containers for our FPAs, where we can use optocells and fly them with inside our gap hardware, which provides the correct containment for samples flying to the ISS. And we can use optocells, which are good for cell culture, and they can either be held statically, where they grow and you don't fix them and they're returned, or this can be set up in a configuration where we can put fixative in them. Next slide. We also have done in the gap hardware Petri dishes, so you can just fly a Petri dish inside a gap container that provides gas exchange, or you can also have no gas exchange. We can also charge the gaps that don't have the gas exchange membranes with the gas mixture that you need. Next slide. And then we have a couple of other pieces of hardware just to demonstrate. We have what's called the BioServe culture apparatus, and it's a smart cell culture piece of equipment where you can grow your cell cultures and then take samples and also fix them. Next slide. And we have another piece of habitat for cell culture as well. It also utilizes the optocells, but it's automated, so it can take samples, put in fresh media, and fix the samples if you need to. Next slide. And they can also be put together in different configurations. Next slide. And you can go to the next one. We also have experience with animal research. We've conducted 5 experiments within the AEMs in partnership with NASA Ames, so we're experienced with flying rats and mice. However, I think, which was mentioned, there currently isn't an animal enclosure that's certified to fly on the ISS. I know the Italians have built the mouse drawer, but I don't think it's quite ready for use, but hopefully over the next few years there will become an animal habitat that is available, and then we have expertise in conducting that animal research. Next slide. And the next slide. We also have developed several small habitats that we've flown many small organisms in, primarily for our K-12 education program, where students in the classroom have flight similar hardware in their classrooms, and then we have a payload operations and command center, so our CGBA has the ability to take images, still and video, and we downlink those to our command center, and then we uplink them to the internet, and the kids can see almost in near real time what's happening in the experiments inside CGBA. And so we've conducted several small organism experiments for our K-12 program, and these can also be utilized by investigators. We've done silkworms, ants, spiders, fish, bees. Next slide. We've done orb weavers, butterflies, and we just recently finished the butterfly experiment. Next slide. And I don't know if any of you saw, we got quite a lot of press on the monarchs in space. And so this is actually a picture of the butterfly habitat. It's inside CGBA, and this has been downlinked to our command center, which then the kids got to see the full development of the monarch butterfly larvae, and then they pupated, and then the chrysalis. Next slide. And these are just some samples of our other small habitat. Next slide. And next slide. And this just shows how the habitats sit inside CGBA, and they sit across from the imaging system. So we have small camera modules to take the images of the experiments. Next slide. So in summary, basically we're a turnkey solution or a one-stop shop. You know, we have a highly experienced team. We have a large inventory of flight hardware available. We're already operating on the ISS. We can modify hardware as needed to help your science get its optimal value from conducting it on the space station. We're fully knowledgeable, obviously, in all the things that need to get done to fly your research on the space station. We have a long, solid relationship with NASA, and We're dedicated to your success in space. If you guys succeed, then we succeed, and that's really what we are all about. And that's it.